This is TWIS. This Week in Science, episode number 1001, recorded on Wednesday, February 5th, 2025. Has the science bell been rung? Ding, ding, ding! I'm Dr. Kiki, and tonight we will fill your head with bitterness, better pain, and active listening. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer. Activism, activists, advocacy, advocates, anti-racists, barriers, biased toward, biases towards BIPOC, Black, and Latinx, community diversity, community equity, cultural differences, cultural heritage, culturally responsive, disabilities, disability, discrimination, discriminatory, diverse backgrounds, diverse communities, diverse community, diverse group, diverse groups, diversify, diversifying, diversity, equity, diversity and inclusion, enhance the diversity, enhancing diversity, equal opportunity, equality, equitable, equity, ethnicity, excluded females, fostering inclusivity, gender diversity, genders, hate speech, Hispanic minority, historically implicit biases, inclusiveness, inclusivity, increase the diversity, indigenous community, inequality, inequitable, inequities, institutional, LGBT, marginalized, minority, multicultural, polarization, political prejudice, privileges, promoting diversity, race and ethnicity, racial diversity, racial inequality, racial justice, racism, sense of belonging, sexual preferences, social justice, sociocultural, socioeconomic status, stereotypes, systemic trauma, underappreciated, underrepresented, underserved, underrepresentation, underrepresented, undervalued, underserved, victim, women, and underrepresented. This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back again.
Hey, what was that list, Kiki?
Oh, just a little open, spoken word, poetry, not really improvised, but actually taken from a list that has been making the rounds on social media that may or may not be part of the list of words that are the words that elicit a review and potential removal of your government funding if you have an active grant with the NSF, NIH, CDC, and other organizations. These are and that's not even the whole list. That's about 10% of the words that are included in the list that have been making the rounds, that have made their way out of NSF. All told, though, they are not verified. Nobody has from NSF, NIH, the government has said, here is the list of words which must be excluded from the government documents at this point in time. But words have been pointed out historically as to what should be used, what should not be used. 2017, for example, might be another year in our past that you would recognize that impacted science. And 2016, 2017, we saw changes in funding, changes in the ability of governmental science organizations to be able to share their information. We saw the EPA, the CDC specifically in 2017 was not allowed to use the term evidence-based in any of its reporting. So words matter. Words are important, and yeah, and science communication depend on words.
And we will not be censoring ourselves here. In
No.
Yeah. But I think also I had a conversation today that I think is really important related to this, which is do not comply in advance.
Nope.
So there's all these conversations about things that may be happening, that may be told, that may be asked of people, that there aren't actually any laws or rules behind these requests. And
Actually, they're
so,
illegal. They are
yeah.
actually not in.
And
Um,
so while
yeah.
you have rights, do not comply in advance. I think that's really important. If you get
Mhm.
asked to do a ridiculous thing as a government employee, then,
About it.
maybe you don't have to comply if you don't know what statute or law or governance is being cited in that request that is ridiculous. So,
Maybe
don't
we
give
should
away
have.
your rights willingly.
I don't know. Maybe there should be like, the constitutional governmental lawyers on call for government employees or for anybody. Excuse me. I don't know if I can comply with that request. I mean, the TPS report, not really fond of that one, but you know, I'm going to have to talk with my lawyer before
Mhm.
I get back to you. No, I'm not. No, maybe I won't respond to this email today. My
Mhm,
paperclip.
mhm. Yes,
Right.
I was speaking to a federal employee who was told, "Don't comply with anything within 48 hours."
And people,
Because a lot
unfortunately,
can happen in 48 hours.
and that is the point that I hope to start bringing up as we get into the stories for the show tonight. I am going to talk right off the top, just so we get past it a bit more about why
Mhm,
we are bringing the science, the politics into the science once again on This Week in Science. And so that is going to be something that we're going to address a little bit more in just a minute. Additionally, I have science stories about managing pain. Maybe there is a new treatment on the way, preventing burnout for your immune system, maybe
mm.
moving ears and bipaternal mice, and maybe some more stuff. What's in the animal corner, Blair?
Oh, that sounds all really good. I have octopus sex chromosomes, starting out strong.
All
I
right, very
have
good.
bat conversations, and I also have frog allergies.
No, you love frogs. I hope you don't have the frog allergy.
I am not allergic to frogs. The discussion is about what the frogs might be allergic to.
Ooh, I'm
Yes.
fascinated in that. Okay. Cannot wait to hear about all these wonderful things. And as we jump into the show here, I want to remind you that, as always, TWIS is available as a subscription podcast. You go to your favorite podcast platform and you look for This Week in Science, look for TWIS and subscribe. Get your friends to do the same. And then you can all talk about these things we're talking about. It's like we're all talking about it together. And oh my gosh, that everybody's talking about it. And just super cool. You can also find us live every Wednesday Pacific time at 8 p.m. On YouTube, Facebook, and Twitch. Our Twist Science streams or This Week in Science are the places you'll look. Our website is twist.org. And now it is time for the science.
Yeah, let's get into it.
All right. So, like I said, we're going to do the politics right off the top so that
Mhm.
we can just dig into why those words are important, why these conversations about federal employees and science and what's happening. Like, if you haven't been paying attention to the science news in this area, in this particular realm for the last couple of weeks, why is it important and why are these words
Mhm.
important and why what Blair was talking about with not complying in advance, why it might be incredibly important right now. Um, well, currently, the White House, Trump has released executive orders mandating the removal of any and all utterances of DEI. Or diversity, equity, inclusivity, and accessibility. And what this means is that um, not just the removal of the utterances of the removal of all departments, the cessation of any programs and
Mhm.
grant funding that has been allotted to projects that are based on these aspects of broadening participation through
Mhm.
society with the sciences. For the last couple of decades, the NSF has, and NIH as well, they have been working. There is a special part of the grant proposal called broadening participation. It's the broader impacts per part of the grant proposals. Every grant that gets funded by the National Science Foundation has to think about how it's going to take its findings and make them accessible, interesting, useful for more than just the people who find those things in a journal article. What are the broader impacts? How is this going to impact society? And so every grant proposal out there has words like increasing diversity of,
Mhm.
or impacting equity, or there's just the idea of removing this stuff from grant proposal language at this point in time is laughable. So in any case, pretty much grant funding is on hold for the large part.
Mhm.
NIH, I believe, has started to put their funding engines back in motion for those grants that are active and are already funded, but proposals are still being reviewed, and that is a question as to how many grant proposals related to community health, related to particular populations, subpopulations of individuals in different areas of the country, Black populations and cardiac health, or diabetes, and there are so many things that we're learning about the very specialized impacts of certain treatments on women,
Mhm. Mhm.
on people
Mhm.
of color, on the combination of all these different parameters, right? So
Yeah,
the idea.
there's three fronts. So
Yeah.
there's making sure
Yeah.
there's diversity in the science that's being done. If you're running human tests in particular, are you covering everybody?
Mhm.
Because that's good science. Okay. Number two is making sure there's diversity in the people doing the science. Because
Yes.
as we have talked about time and time
Pipeline.
again, there's someone will go, oh, what about XYZ? And the whole white male population that had done science previously is like, oh, I never thought to ask that
Yeah.
question. Right.
Yeah.
Um, Similarly, things like Black researchers, the automatic lights turning off on them in labs because those
Mhm.
things are oriented to white people, right? But then three is the piece that you mentioned at the beginning, Kiki, which is making sure that the science information is reaching everyone.
Yes.
So it has to be part of it. It's part of good science. It's
Yes.
the fact that we're just, over the last couple decades figuring out that there were these holes is terrible. And it's excellent that there's been a really intense concerted effort to rectify those blind spots.
Yeah.
Um,
And it's been working, and we've been building up the knowledge. We've been reaching more communities within the United States and outside the United States. I mean, honestly, what we do with science communication depends on the ability of researchers to talk about their work, of different departments within universities and the government and different organizations affiliated with those, to be able to communicate, and then to be able to communicate with the communities that they are connected to in the world. The restricting of communication, the censorship of communication, is going to simply reduce the knowledge and the welfare of society.
mhm,
Science is the knowledge generating engine for society. And if we are not a part of it and aware of it and what it's doing, it will not be working for us. And where's the pipeline? How are people going to be
mhm,
interested in it? So
mhm,
anyway,
mhm,
yeah, all the things that you're talking about, Blair, this is the three pillars. You're exactly right. And unfortunately, right now, what we are looking at is a situation where researchers are unable to get funding. They don't know where they will get their future funding because of the things that they do study, depending on how that's going to work out. Labs are kind of in turmoil, which is in
mhm.
turn going to impact universities, which take a 60% overhead off of all of those NSF and NIH grants, just for all y'all to put that into a little bit of understanding of your economic budgets of universities and where grant money goes. And that's to pay for the labs, the facilities, the people, the administration, all of the things. Beyond that, we have questions of how are we really going to engage society, our populace, our public? How is this going to move forward? And as the Department of Education is potentially being shuttered, the CDC is being hamstrung. NASA, what did NASA even like is shutting down like its little kid award programs right now? Like I'm so sad. And there is a certain amount of turnover and stoppage of work and various review processes that occur at the transition of every presidency. So what is old is new again, but is this that just that transition or
No.
No, I don't think so either.
No. It's also not a coincidence that funding sources for colleges are being shuttered at the same time that the Department of Education is under fire. That is not a coincidence. That is part of the
break it all down.
Yeah, the plan to put everybody in the dark as to what's going on. So we need to keep each other informed. I know there are kind of alt social media accounts that are working to keep people informed. There are people posting data that got pulled from the NIH website. There are people trying to keep bird flu information out in the populace. So like there are people trying to help, which I think is somewhat, hope inspiring. There are glimmers of hope here. There is resistance. There are people who want to keep information out there at
There's the entire scientific community.
Yeah. 100%. So people want to help. I think everybody has some whiplash right now because it's all happening so fast and so brazenly and in channels that are not legal, which makes it complicated.
And that's the 48 hours thing. So many things can happen in 48 hours if you don't know the legality, where it's coming from down the chain. Who are you supposed to listen to? What are you supposed to do? So
but so I mean,
Pepsi Cola is saying lawsuits and a lot of people and organizations, there are things being filed. And so the legal system is in process and hopefully our judicial system works the way that it should. And it maintains the limitations on the executive branch and allows for the, continued limitations on, or the boundaries between executive, judicial, and our Congress.
But I think something also to keep in mind with all of this, because we hope that the checks and balances hold. We hope that some of these ridiculous things that are happening get pushed back. Some things have already been rolled back or rescinded, or all sorts of things. So I think everyone needs to give themselves some grace right now, because this is engineered to give you panic. This has been created in a way to make you feel hopeless and like manic and like everything's happening at once. That's intentional. And so the best thing that you can do is take care of yourself, take care of each other.
Breaks. Yes.
You might want to go march in the streets right now, and if you can get that together to do that, that's excellent.
During one of the protests, that's fantastic. But if not, it's okay. Do what you can do. You can do it.
Because in terms of organized action, we might need a minute to figure out what's actually happening. Is this all a distraction for something else that actually needs our attention? So I think it's so early and it's so overwhelming, and that is intentional. Like, take the time to figure out what's going on, to see who needs our help right in this moment,
and figure out if you're Michael Jim Burns is saying that there's a gathering, there's a lot of people gathering on Blue Sky. I love Blue Sky. I follow journalists and scientists and science communicators over there. And I really think the caveat there, as with social media all the time, is figure out who you can trust and take a minute, stop the doom scrolling because the emotion of the things people are saying because they're getting it off their chests, they're angry, we're scared. You know, those are real feelings and valid feelings, but if you keep scrolling, you're going to potentially emotionally react to things
Mhm.
that you should take another minute to consider more rationally. Where did
Mhm.
the remember, where did the source of that information come from? The reason I said at the beginning of the show that the words that I said they are purported to come from the government as part of the banned words or non-governmental use words anymore, but there's no real source on that. It's like a bunch of hushed, whispered things,
Mhm.
and somehow these words have made it out. So um, just remember to watch yourself, check your sources, check the timeliness. Is this from 2017 or is it from now? You know, make sure you know who you're talking about. Is this real even? Is it
Mhm.
manufactured? It's because there are trolls, there are AI bot farms that are,
Hmm.
you know, s- pushing out disinformation all over the
Just
place.
throw
So
that in the mix. Jeez
yeah,
Louise, makes that more complicated
it's
than
complicated. So everything, I just want all of us, even with this show, right? Tell us when we're like reacting too strongly. But I think you're right, Blair. Though there is there are other people. Take care of yourself. Watch your information and know that you're not alone if you
Mhm.
are feeling overwhelmed. And remember that science is a generator. It's a method. Science won't go away.
Mhm.
People will
Mhm.
always do science because science is part of how we create our future. So it's just there that is going to be fought for because it is so important. Yes, and yeah, ex-MAGA, we must find time to stop and thank the people who make a difference in our lives. We can complain because rose bushes have thorns or rejoice because thorns have roses. I think that's wonderful. Thank you for that sentiment. I really do appreciate that. Because, yeah, and remember, these things that I'm talking about, the psychology, the stopping for gratitude and reframing stuff for doing this is all science, evidence based
Yep.
information about your brain.
100%.
We're not
Absolutely.
just throwing stuff out there.
You can't live in fight or flight you're all the time. You biologically cannot.
Oh, come on,
So,
Blair. Come on. I could totally do it.
well, see you in the hospital next week.
No. Nope. Not going to do it.
No.
Yeah. Anyway, there's a lot that's happened in the last 2 weeks. People are in the sciences are uncertain, but this is an early period of time. And like you said, Blair, this is going to play out. And for all of us, um, things might be hobbled a little bit for the time being, but hopefully they will come back even stronger. What do you want to talk about today?
Oh, do you wanna just talk about frogs?
I want to talk about frogs. Can we talk about
Okay.
frogs?
Oh my gosh,
Let's
I
do
love you. That would be amazing.
it. Yes, please.
So, did you know that frogs are basically covered in taste receptors?
I do remember you saying that at one point in time, which was I had forgotten until you just reminded me, but
So,
now I am just thinking of frogs as
as a
weird,
giant tongue?
tongues. Yes.
Yeah.
Yep.
Yeah, well, because they also breathe through their skin, so it's just a permeable membrane. There's a lot going on there with frog skin, with amphibian skin. And so researchers from Northeastern University wanted to look at bitter taste receptors known as what I'm deciding to call tasers, but it's TAS2R plural. TAS2Rs. Um, those are the bitter taste receptors. We have 25. Most of ours are in our tongue. There's some in our gastrointestinal tract, and there's some in our brain. So pause. Kiki, why do we have bitter taste receptors in our brain?
So that we can sense our bitter thoughts.
See, I really thought you'd know. I didn't look it up because I was like, he'll
No,
know.
so bitter receptors are important for the sense of toxins, for sensing toxins. So any place that a bitter receptor is very likely going to be binding to something that's potentially toxic. And now we kind of like bitter things, but once upon a time when we were evolving into where we are now, it was like, ooh, that bitter thing, maybe if I eat too much of it, it's going to make me go bleh and vomit or die. And
Oh, okay. So the
so
receptors
the
are in
in
your
the
brain
brain.
to basically talk to the receptors in your tongue? Is that
Mhm,
okay?
mhm.
Got it. All right. So and then of course they're
Toxins end up in
also
your brain, then it's a receptor and it goes, "Ah, protect."
got it. Got it. Got it. Okay,
And
that
the
makes
immune
sense.
system can get involved.
Okay. Excellent. And then the GI tract is so you can purge from the other end if you need to from toxins. So that's fun.
Mhm.
So we
All of
have
the
25.
above.
We have 25 bitter taste receptors. A wood frog has
I would have guessed I had more.
248.
248?
Yes. So some are in their liver, and a lot of it
Mm,
is in their skin. And when you eat something bitter, you spit it out or you poop it out, and that's the bitter receptor is kind of cueing into that. So what researchers wanted to see was how the bitter receptors work hand in hand with the presence of toxic proteins to activate immune system responses, and specifically if those receptors are related to responses to allergens. So, is the receptor responding to an allergen, and that is why you're having an immunoresponse to that allergen? Basically, are they mistaking that for something that is should be attacking your system or should be toxic when it's not, right? Um, there's still a lot to learn about how the mechanism of how these receptors work, but the researchers looked at a database of 680 species to look at their receptors. This is a huge study. They found a surprising evolutionary story. The amphibian family tree has an explosion in the number of bitter receptors. The boneless fish that amphibians came from had, depending on what you're looking at, either 1 or 0 of the TAS2Rs, or I'm calling them tasers. But in frogs, the number ranged from 50 to 300. And so this appears to be an evolutionary adaptation specific to frogs. As an example, dolphins have 0 because they eat non-toxic fish. But what do frogs eat?
toxic fish.
They eat bugs.
Oh, right.
Bugs have been around for a really long time evolutionarily, and amphibians and bugs are in a bit of an arms race, chemically speaking, where a lot of bugs end up being toxic to try to avoid being eaten by amphibians. This is a push-pull that's been going on for millions of years. And
And isn't it
so,
true that there are some of the amphibians, the frogs, that will take the toxins from the insects and use them themselves to be more toxic, like concentrate
yes,
them? Hmm.
yes, that is what poison dart frogs do.
Mhm.
And poison dart frogs, if you ever see poison dart frogs in a zoo, they aren't poisonous because they haven't been eating the bugs that make them poisonous. They're just eating
Hmm.
plain feeder bugs, and so they don't have any toxins on their body.
This is
So they
good to
still,
know. Yeah.
yeah, if you wanted to lick a poison dart frog in the zoo, please don't.
Or, go hunting with arrows? I don't know.
Sure.
Whatever keeps you
Yeah, yeah, don't rub the zoo frog over your arrows before you go hunting, for
no.
sure. Um,
Uh,
So, poison dart frogs. They've developed a way to make their own toxins in after having ingested them. So, detecting how much toxin is in something they're about to eat is still valuable information because they're seeking out the toxic bugs. Similarly, in some of the other species, they have a huge concentration of these receptors in their liver, which helps detect toxins when they enter the body, which triggers a process that metabolizes them. So there are other frogs who can eat poisonous bugs because they have the receptors in the liver to go turn on the poison synthesis, right?
Or the metabolism to break down the poison and turn it into something else.
Right.
Yeah.
Yes,
Yep.
absolutely. And so basically what they've learned from all of this is that frogs are unique in the evolutionary tree, having this huge explosion of bitter taste receptors. And based on how they respond to toxins, they think that it has a role in the way they perceive antigens as well. And so that
Mm.
means that it could be related to immune responses, which means it could be related to allergic responses. And if they can drill down on that, then that could be a treatment for allergies in humans. It's a bit of a long walk,
It is,
but I
but
see where
it's,
they're going.
yeah, step by step, right? Just put the pieces together and
Yeah.
suddenly that long walk, it's a short pier. No.
Long walk up a short pier. No, we're not doing that right now.
Okay.
Not unless you're an aquatic frog, I suppose.
This
Yeah,
is pretty
so
cool.
so frogs and all of their bitter taste receptors all over their body and in their livers, they could help provide treatments to allergies in the future. Stay
That is
tuned.
fascinating. Oh my gosh. I wonder though, yeah, I guess it's because they're constantly in that, like you said, battle against the insectivora, the class that they eat. To be able to
Mhm.
sense the bitter, they have to be able to sense the toxin so that they can respond to it, whether break it down, use it, spit it out, whatever. Um, you know, I guess we don't eat enough bugs.
Yeah.
Only 25 bitter receptors? We're so limited. Jeez.
Oh,
We know nothing. We know nothing.
mhm.
That's all.
Eat more
It's
bugs, I
fascinating.
guess. No,
I love the
don't do that.
connection between all the different organs in the body as well and
I
how that
know.
works.
That's very confusing to me that we have bitter taste receptors in all those places. It's wild.
Because it's a receptor and it
Yeah.
kickstarts processes, right? Oop, there's a
Mhm.
thing here. I recognize it, and it's toxin. It's bad, bitter, toxin, bad, or whatever. Mm.
Mhm.
But that's not the only thing we need in the world. Just don't go licking frogs unless you're at a zoo. Um,
Go licking frogs. Nope.
no, no, I'm not going there. Uh, this week, for people who suffer from various kinds of pain, particularly those individuals who have had acute lumbar pain. The news out of the U.S. Food and Drug Administration as of January 30th should be pretty exciting for you. This is the first approval in a very long time of a pain medication that is non-opioid, non-addictive, um, and it's been more than 2 decades since a new drug class has been approved to treat acute pain. And acute pain is identified as less than 3 months of pain. If you're going on after longer than 3 months, that's chronic pain. So this particular drug is called suzetrigine and is being manufactured and is going to be sold under the name Journavx by Vertex
Catchy.
Pharmaceuticals. Yes, exactly. Um, and this new novel mechanism is actually really interesting because you know how we have sodium, we have voltage gated sodium channels in our nerves. And a lot of neurons, it was discovered during a study of people who don't feel anything, who have no sensory, no pain feeling at all. And the other end of the extreme, which is called the man on fire syndrome, where the whole body, the skin feels like it's burning. Um, they were able to discover that there are pain sensory receptors and neurons in the process of pain perception, that are these receptors are little sodium channels in the membrane of the nerves. And these sodium channels, they're not turned on by touch, they're turned on by voltage. And so neurons are activated by what are called action potentials. And so you have this switching of chemical gradients across the membrane of the neuron. And so a signal comes in, and action potential, and it changes the voltage potential of the membrane. And when that gets to one of these voltage gated sodium channels, dun dun dun, you have an opening, sodium flows in, potassium flows out, or maybe it's the other way around, but it triggers a shift in the internal environment of the cell that then either starts another process or perpetuates the action potential. And so they've discovered there are a whole bunch of different kinds of voltage gated sodium channels, subtypes, and they've numbered them. And so they've discovered for this particular mechanism, there's these very conveniently numbered the voltage gated sodium channels 7, 8, and 9. Um, Voltage gated channel 7 is like the low volume one where it's like, oh, little gets stimulated a little bit and it goes, oh, that's something, that's interesting. And so it turns on, and then it gets bumped, and it hits the number 8 voltage channel, and that one goes, ooh, this is exciting, and it turns on a big signal. And if it does a big enough signal, then it triggers the number 9. And so number 9 keeps it going. And they've actually been able to identify that this triumvirate of dominoes that fall down along this pathway is a really amazing target for treating pain. And so they now have this new drug. It's gone through 2 different phases of testing within the Food and Drug Administration, and it is on its way with almost 1,000 people having been tested so far and having fairly reasonable pain reduction measures, and having very limited side effects as well. So some people have had headache as a result, which you're like, oh, I took a painkiller and now I have a headache. But they're with the number of people they've looked at so far, it hasn't really had a lot of negative side effects. So phase 3, we're getting into the large scale trials. And if so, this is really a big part of actually the government's push, which was opened up this January 1st, the Non-Opioids Prevent Addiction in the Nation Act that became effective as of January 1st, that mandates Medicare to provide a separate add-on payment in outpatient or surgical center settings for FDA-approved non-opioid pain therapies. And if this moves on as successfully as it has so far on its studies to date of looking at, they've done, let's see, phase 3, they've looked at surgical and non-surgical participants. They have looked at orthopedic, plastic, otorhinolaryngologic, I can't say that word. I try to say lots of words and sometimes they don't come out well. Non-surgical cases, sprains and strains, and they determined that 83.2% of the participants rated suzetrigine as good, very good, or excellent for pain relief. And now it's moving forward as well with painful lumbosacral radiculopathy treatment. And all of these things, if you're not having to give people opioids for the pain treatment and you're giving them something that is, it's non-addictive, it's just going to work when you need it, this is a huge step forward. So fingers crossed, but FDA has said, move on ahead, keep going. This train is exciting. Wait, I don't hear you. Where'd you go? You're muted.
Oh, I said 1,000 is good, but I'd sure like bigger numbers. So I'm glad to hear that it's been given the go ahead to proceed.
I mean, that's very often when things hit the larger numbers, they really do make it. But this is where it really is the make or break, right? But we've gotten past this drug, this treatment has gone through a lot of the major hurdles to get to this point.
A new painkiller is we haven't had that in a while, I feel like.
2 decades.
Yeah.
It's ridiculous. It's amazing.
Mhm.
It's amazing. I love it. This
Me
is and this is a long-term process of research that has allowed us to get to this point because you can't just like it's one of the earlier forms of the sodium channel treatment is Novocaine. So the but that's like you have to it's topical. It's a cream or a lotion or something of that sort, right? Or an injection. But this is something that could be like an ibuprofen or something. It'd be a pill,
Mhm.
and you could take it home, and it would be internal and not topical. And yeah, could be really great. What do you have
Neat.
to look forward to, Kevin Reardon? Less pain.
Mhm.
And then for the last little study for this particular part of the show before we move forward into the animal corner, another exciting advancement this last week. Researchers from the Melbourne-based Peter Doherty Institute for Infection and Immunity and the Peter MacCallum Cancer Center, they have identified a rare type of T cell. It's an immune cell.
Mhm.
You may have heard of T cells in the the fight with of our body against HIV/AIDS, and T cells also are essential for so much of the treatment of chronic diseases. And very often these T cells, they go for a good long while, but then they just kind of they can't replenish themselves. And so researchers, what they've found is a T cell type that's like a stem cell. It can rebirth, it can regrow, it can reinvigorate its population, which means that treatments like CAR T cell therapy for cancer and other treatments where you go in and you reprogram the T cells, eventually these treatments they stop working because even though you have taken out T cells, you know, maybe done a bone marrow transplant, and you've taken out the T cells from the old, and you've got these new reprogrammed T cells in. Eventually, they wear out, and when they wear out, you've got these you have a lack of response, or you have go back to the same old response again. And so, in this situation, by finding these stem cell like T cells, if they can figure out what allows them to have that property, the stem likeness, it could maybe allow researchers to be able to adjust the CAR T cell therapies to create longer lasting results, or maybe results that are constantly reinvigorated on their own. So it's I think, a very
Hmm.
exciting. I mean, I would like personally to be reinvigorated every once in a while.
Hmm.
I am definitely not STEM like, but that's and maybe after a good night's sleep or
What's that?
a week sitting in a chair by a
Oh
river
my goodness.
reading
Oh, that
books.
sounds great.
Doesn't that sound lovely? I am dreaming of summertime or already.
Hmm.
Summer, summer, summertime. Science. Yeah, but it's this kind of discovery that is very exciting because it has the potential to um, allow us to really change the way that we treat and manage chronic diseases like HIV and diabetes and other infections and cancers that are long lasting that the body needs to regenerate and reinvigorate for. Also, I think because of the podcasting thing, I don't know, this study really like the ID3 plus T cells. It's like the T cells have ID3 tags, but anyway, that's old school RSS
Hmm.
information. But it's kind of the type of cell is inhibiting DNA. What is it? It's the ID3 is inhibitor of DNA binding 3. So it's a protein that
Mhm.
inhibits certain DNA binding. And in having this ability to resist the DNA binding of certain kinds of binding, um, they are allowed they keep signaling, increasing their numbers, and they keep
Hmm.
going. So it's very exciting. Yep.
Nice.
New methods for fighting the diseases.
That's great. I'm glad you had some good news for us.
I try, right? There's got to be. That's what we're here for. We're here for the good news, for the positive. Let's look at the optimistic side of what science brings up us, everybody. That's what I've always enjoyed so much about this show. Blair brings us frog allergies that might help save the world. No. Bitter frogs that might save humanity from allergies in the future because, allergies are getting worse. We need that. And we love frogs. Save the frogs, everyone. And, I have to bring some positive future thought of how we will, how we will live better lives. That's what science, the hope that science gives me every single day. And especially every week when I get to be here talking to Blair, when Justin's here. Justin, and all of you, thank you so much for joining us for another episode of This Week in Science. You can find more information about the show at TWIS.org. Please share This Week in Science with other people you think will enjoy all this science conversation. We're going to delve deep into some more science here in just one moment. And in the meantime, if you're headed over to TWIS.org, you can click on the Zazzle link at our website, and that'll take you to our merchandise store where you can find all sorts of cool TWIS gear and what lovely pieces of artwork on towels and shower curtains and t-shirts and bags and so much that Blair has created through the years, and it'll help support TWIS and also make you look super cool and sciencey and artsy, arty for sure. Also at twist.org is a link to our Patreon. So if you would like to support us in an ongoing fashion, click on that Patreon link and head over to Patreon, $10 and more a month, and I will thank you by name at the end of the show. And really, we're not government funded. I can say female diversity women as much as I want.
DEI.
Oh, no. Now we're a target. Thank you so much for your support. Thank you for being here. And without any further ado, I will now bring us back to This Week in Science with Blair. It's time for that part of the show that we have always known and loved as Blair's Animal Corner with Blair.
She loves our creatures, great and small. Biped, milliped, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Octopus sex chromosomes.
So do they keep those in like one tentacle or all the tentacles?
Great question. They're everywhere, man. So sex chromosomes. We think about sex chromosomes in humans as the XY dichotomy. Very apropos with the conversation we were just having. In humans, the XX is female and the XY is male. But it's not as clear cut as it might look on paper. Sometimes there's mutations. Sometimes people have multiple chromosomes. Sometimes they inherit an extra chromosome that leads to development that doesn't fit into the male-female binary. So like the male-female binary at birth is actually kind of not 100% true. But ultimately, we have a chromosome-based determination system, depending on, in the grand majority of cases, if you have two X chromosomes, you are born female. If you have an X and a Y chromosome, you are born male. Alternatively, turtles have temperature-dependent sex determination, as do crocodilians. Yeah. And so, depending on what temperature the eggs are incubated at, then it might hatch a male or a female baby. So different animals have different sex determination methods. And also some lizards, like the Komodo dragon, they have what's considered a ZW chromosomal system. So the heterochrome of ZW is actually female, and the males are ZZ. They're the homochrome, as opposed to
They're the
how
top.
we. Yeah, so they're the opposite of what we have. So basically, I say all this just to illustrate that the XXXY situation is not consistent across the animal kingdom. Animals do all sorts of things to identify the reproductive equipment that they are born or hatched with. And so,
And I want to
yes.
just bring up some Pepsi Cola is saying octopus and platypus are such strange and complex creatures. And the platypus and the echidna I love so much. And just remember that they are like, they have like 9. It's XXY, XXXXXY. Like they have 9 sex chromosomes. 9.
Yeah.
So come on.
Yeah. So the way that it works is not
there.
universal. It's all over the place. And ultimately, for octopuses, we've never really known how they identify, how they get the definition of sex at birth.
It's because they don't have name tags.
Right. So also, it can be kind of hard to tell what is a male and female specimen when they arrive in the lab. The easiest way to tell is via if they have a hexacotylis, which is a section of one of their tentacles that is missing suckers, and the male uses that to hand a sperm packet to the female. Very cute. Anyway, that's the easiest way to tell if they're missing it, if it got bit off by something in the ocean, if the specimen is not well preserved and everything's kind of squishy. There could be a million reasons that you wouldn't really know. And also, historically, genetic samples have been collected and then not labeled as whether it was from a male or a female specimen. So difficult to do historical research on collected DNA specimens because you don't know what you're looking at. But this week, University of Oregon researchers identified what they think is a sex chromosome in the California 2-spot octopus. So what they did is they sequenced the DNA of a female California 2-spot octopus, and they found something unusual, which is a chromosome with only half of the amount of genetic material. It looked different from all the others. It hadn't been found in male octopuses with the DNA who was previously sequenced. So they found this thing that was absent in the male, and they only found half of it in the female. And so there are 2 options here. Either they have the kind of ZZZW determination that we talked about in birds, butterflies, and some lizards, where the males have ZZ and the females have ZW. Then what they're seeing is that W, and it's only present in one copy because it's a heterochromal definition of. Does that make sense? Am I losing you? You got it? Okay, great. Okay.
Hetero, different. So there's like
I've been explaining science all day.
Yeah.
I'm losing my touch. Okay. So okay. So because they only have the one copy of the chromosome, either it is that system where the females have the 2 different versions of the chromosome that identify them as a female, or the other possibility is that they could use a sex determination system that only involves one chromosome, like the Z chromosome. Males would have 2 Zs and females would have 1. That is the other option. So those are kind of the 2 hypotheses they have. And moving forward, there is a lot of research to do, because this is the other thing that's really, really interesting. They found another example of the half-size chromosome in another species of octopus. Okay, so backing up evolutionary time, all the octopuses have it, it would appear. Then they found it in a squid. Squids diverged evolutionarily from octopuses between 248 and 455 million years ago. That is a long time ago. And then as they continued to research, they also found it in the nautilus, which the nautilus is this like basal cephalopod. So now you're backing up evolutionary history to at least 480 million years ago. This is an old sex chromosome.
And they held on to it.
Yes, and that is really unusual. Sex chromosomes are generally very young in evolutionary history, and it's because they're directly related to reproduction, which is the mechanism for evolutionary change. And so because of that, it is often not retained. The kind of chromosomal definition of sex is not retained over evolutionary time because selective pressure, right? But somehow these guys have kept it for 480 million years.
It's because they had a lot of tentacles.
Yeah. Grab on.
But I mean, when you think about that, I mean, some of the octopuses that we have discussed, their mating habits are so unique, obscure, like females that go years and years and years without reproducing, or they hold they have it all inside them until like the one right moment for they're in the just right temperature water to lay the eggs and to have the pods be produced. There's so many unique adaptations throughout the cephalopod kingdom, but to know that they, even with all the adaptations that allow it to work in the various ways for the different species, that this is conserved suggests that it is something that has really, really worked. Right? Yes, Eric Knapp. Absolutely. Well, if something works, exactly.
Yep. Yeah. So I expect to see more about this. This is something that obviously has not been cataloged or kept in labs previously. Also, cephalopods are not super common lab animals, so there's not a huge catalog of research on the DNA of octopuses and other cephalopods. But I do expect to see more, because studying sex chromosomes is very interesting and a very rich field of science, because it is tied directly to reproduction, which is tied directly to mutation and evolutionary change and all selective pressure and all of that. And so it's a very important thing to look at when you're trying to piece together evolutionary past and project out towards future as well.
Yeah. I wonder, so where was this study out of again?
University of Oregon.
University of Oregon. Okay, cool. Interesting. I'm just thinking of all the different places. Monterey Bay Aquarium. I'm mostly West Coast because that's where we are. And you've worked in the aquarium business, Aquarium by the Bay in San Francisco. But the places that have cephalopods, so Cal Academy of Sciences, Monterey Bay Aquarium, probably the National Aquarium in Baltimore. All these places, I'm sure they're the top runners on having the databases or having the resources to gather these the chromosomal information.
Yeah, but now I think the really important piece is they know what to look for. It has been sequenced. It is expected to be a sex chromosome, and now they can start looking for it, and they can start looking at mechanisms, finding out exactly how the dichotomy works. And also, is it related to things like senescence after they lay eggs and then they die immediately in some octopus species? So, there's a lot going on that, it's a potentially very rich field of study. So, I don't know. We'll see.
What will we learn of the cephalopods? What will we learn?
Well, I can tell you what I learned
batty
from bats this week, is that baddies can be chatties. Does that work? I don't know.
Yeah, fair enough.
Um, basically, what was learned this week from a team of animal behaviorists at Leibniz Institute for Evolution and Biodiversity Sciences, Museum for Naturkunde, and Wageningen University and Research. It's German, all German universities and research centers, I believe, has found an association between different types of vocalizations in Pallas long-tongued bats and certain behavioral characteristics. The one sentence version of this study is extroverted bats talk more.
I'm surprised. No, I'm not.
I mean, I'm not going to say I'm surprised. I was going to say never heard this study before.
No.
So here we are. They studied
Okay.
60 specimens in the lab. Prior research showed that bats vocalize, all species, but some more than others, and some individuals more than others within those species. Prior research also showed that most bat species exhibit social behavior through movements, through vocalizations. And so in this study, researchers looked at the role that vocalizations play in social behavior in these Pallas long-tongued bats that lived in a cave in Costa Rica. So they took 60 individuals, brought them back to the lab. They were all males, just FYI. I don't know. Does that matter? I don't know. But that's what they did. They were exposed to novel
Probably.
experiences like a new environment, a rubber ball, food next to a lantern, and then bats were recorded as they responded. So they were able to study physical behavior and vocalizations. They found that bats who behaved boldly during the experiments vocalized more. Bats that showed more curiosity towards the rubber ball were more likely to sound an alert if they deemed one necessary. They also found that bats were more likely to vocalize if it appeared that they were more nervous during the experiments. So, and they responded by moving around more and flying for longer periods. So they were just generally more active. So this suggests that there's a difference in vocalizations and behavior related to personality. They also suggest that differences likely have an impact on social hierarchy, but they did not do that research yet. It's kind of an extrapolation. And we know there's a hierarchy with bats. We know that prior research has shown that bats eavesdrop on other bats, for example. So it would make sense that social hierarchy and social dynamics would be related to these personality differences, but that has not been actually experimented yet. There hasn't been data on that. But ultimately, yes, more adventurous, more nervous, more active, they're going to talk more. So just generally heightened. Oh, you're muted.
I said bats that are more are more.
Yes, exactly. Bats that are more. You got it.
So what I'd love to know is with the personality differences, I mean, this is obviously adaptive, right? So do these individuals do like in the case of bat social hierarchies, do they have more individuals that will help them groom, that will be potential mates? Do they have larger friend circles? Do they catch more insects? Are they louder? Are they more extroverted? Are they actually better at being a bat friend? Or are they
Great
just
question.
louder and do they just talk more?
Yeah.
Like
Yeah. So that's really, I think, Kiki, you hit on something really important. Are these personality differences or is it fitness differences?
Yeah.
Is this them being more bat is better? Is more bat worse? Or is it just personality differences? Because I think that's really the key. Is it just all bats are different? There's not really a positive or a negative to it. Takes all kinds. Or really, if you are playing the fool and you're out there and you're making sound when you shouldn't be, are you going to get eaten or stolen from or whatever? Or by being louder, are you going to get more help? Right. So,
right. And I'm sure this is, the cost benefit balance, right? Where you have different, different numbers of these different personalities within any population, more introverted, more extroverted, somewhere in the middle. And the balance probably has to do with that benefit of, okay, catch more insects, have more social friends, but also, ooh, more visible, could die more easily.
Mhm. Mhm.
Or
Mhm.
actually are a little bit worse for the group because of the loudness. Or, maybe there
Yeah.
are certain actual social aspects to that as well.
Yeah. So I think that's ultimately my takeaway from this story. Bats have personalities.
I
Do What does it mean? We don't know. What's the benefit? We don't know. But
don't
bats
know.
have personalities and they are predictable based on they could kind of pick out things that bats were doing or ways that bats were acting, and based on that extrapolate if they would be nervous or if they would talk more or any of those sorts of things.
I think this is one of those interesting questions because we have been talking about personalities of different animals, different species, on the show for years, right? So we have these measures of, we could talk about personable pandas, or, we can talk about aggressive squirrels. We can talk about, shy woodpeckers, or, people have been studying animal personalities as a feature, of their um, of how they interact with their environment and their conspecifics for decades. and
Mhm, mhm.
and so just like people, what is it, why do we have different personalities? Why are they definable and or categorizable?
Yeah.
Why are we so easily categorized? I don't know,
That's what
Blair.
humans do. We categorize.
Ugh, are you a lumper or a splitter? I don't know, you're indefinable.
Sometimes one, sometimes the other, you know?
I have no idea what you're talking about. Yes. But she's got personality. Personality. All right. I've got mice two dads. Yes. And I did.
Yeah.
I, do you remember that show? My two dads?
My two dads,
Um,
yes, of course. I also remember Two Guys, a Girl, and a Pizza Place, but that's neither here nor there.
There's that too. Yep. Anyway, new study out in Cell Stem Cell this last week. Never done this before. Never been able to get a mouse with two fathers. That's right. A mouse fathered by animal cloning using embryonic stem cells, induced pluripotent stem cells, and basically taking um, sperm only and creating a mouse. Historically, all attempts at creating a mouse with two dads has ended in the embryo dying at some point or not making it completely to term.
And we
This is
got
the
all the way to mouse?
adults, adulthood.
Oh
I'm going,
my goodness.
yes, this is the first time that this has happened. And what they did in the process to allow this to occur, and I'm going to caveat this here, which is they, this is not perfect. This is absolutely in no uncertain terms not the woo, we did it. We made it. This is it. Guys, we got it. Like, dads only from here on out. No. No, 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 no. The caveats are that a very large percentage still did not make it through uterine development. A large percentage also died as young mice, and the ones who did make it to adulthood had had shorter lives, and also had some disorders. So they weren't
Mhm.
completely healthy, absolutely 100%, the standard adult mouse. But this is the first time that we have gotten to this particular place. And the way that it has happened is through the use of a process called imprinting. And in development, in the process of putting the egg and the sperm together, at a certain point in every single cell, there is a choice. Because every single cell, it's got the sex chromosomes in it, and the cell has to choose: am I the if which one of the sex chromosomes am I? It's which half of the chromosomes am I going to pick? And that is the imprinting process. And each cell can be different, but there are specific markers that are involved in allowing this process to occur naturally, normally. These are all, words that have implicit biased meaning, but in a more normal way. And so they were able to take embryos from same-sex parents, and through imprinting, what they did is they had regulatory edits at 20 key imprinted loci, so spots in the chromosomes, and they achieved, and this is what allowed them to get the fully adult animals. They made sure that there were also frame shift mutations, and there were gene deletions that were important, that they saw as bringing deficits that led to developmental issues. So they learned from previous experiences how to make the process work better. And this is an iteration that could potentially lead to embryonic stem cells really working for reproductive issues and the cloning of animals. So regenerative medicine, we don't just need sperm. I mean, we don't just need eggs and sperm, right? Maybe now, maybe we could just take your skin. We'll take your skin and turn it into sperm, and then we'll take the sperm, and then we'll put those together, and then you got a little baby that's not you. But anyway.
What did they use as an egg, if not an egg?
Right. So it was they took a sperm. They took one of the sex cells of one of the gametes of a male, and it's a different male than the other. So it's not just two sex cells from one individual, because that could induce mutations that would be deleterious. Right. Yeah. It's because sex reproduction, like you mentioned earlier, we want that recombination. Mm-hmm. That's the stuff that swaps things around, and it adds the excitement. That's why
inbreeding is bad, is because you're not getting enough recombination. Yes.
Yeah. So in those early phases, they had early sperm. One of the sperm was then induced through pluripotent or induced to be. No, I'm going to take this back. Rolling it back. Okay. Embryonic stem cells. They have the embryonic stem cells turned one into sperm, one into egg, both from male individuals. So they induced the embryonic stem cells to become one of each sex cell type.
But you're using two sperm.
No, so I got that wrong.
But the diagram, the diagram says first sperm and second sperm in it.
Yeah, it does say first sperm and second sperm. Gene editing.
I'm so confused.
Okay, this is okay. No, okay, here we go. Now,
thank you.
Now we're backing up, rewriting, and starting. Okay. One sperm is just the sperm, right? It's going to be the cell that is used, the sex cell that's used. The other is a sperm in combination with an embryonic stem cell that's haploid. So they've caught it halfway through its dividing process. So it has half the complement of genetic information, and it's big and has all the cell stuff like an egg would. So you have a haploid embryonic stem cell with a second sperm that's put into a little thing. So you have a sperm thing and you have a fake egg thing, and then they get put together. And so you have a haploid. So the first sperm is basically, we're going to put you in a cell and oh, hey, sperm, you've got your own nucleus. That's awesome. Look at you. You've got energy. That's great. We're going to put you in a cell. We're going to take the original nucleus out of that cell. Boop. Bye-bye, body cell. That's great. Nucleus goes away. So now you've got the sperm taking over the cell. you tell it to divide. It turns into a little blastocyst, divide, 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 except it's only haploid, right? Because you've taken out the nucleus and you only have sperm DNA. So you've got a haploid group of embryonic stem cells. And then you edit it and make sure that they are not mutated or strange. And then you have your second cell, and you're like, oh, we're going to add embryonic stem cell with another sperm. And it's put it together, take out the nucleus. So you've got your thing with only one nucleus. You let that then divide, divide, divide. And you say, go be a being. And it eventually, because of the way that they've worked through it, have allowed it to have viable offspring. Another side note is all the surviving offspring into adulthood, all of them were sterile and male.
Okay.
So there were no, it was very interesting cloning. There were no sexual differences because it's all the male lineage all the way down, mammalian male, all the way down. So all the offspring were male clones to a degree, and sterile, and had lots of other issues.
So,
but this is the first time we've gotten living organisms like that long.
Right.
Mice lived, yes.
And I'm assuming that the mouse was incubated in a female mouse, of course.
Yes.
So, but that means that
Yes.
mouse, that baby mouse, really had 3 parents, because the act of carrying a baby also impacts their DNA, correct?
Yeah, the epigenetics. Absolutely.
Right.
Because and that is something that is not discussed. I mean, it's just, these are lab mice. So
right. But just to be clear, if we did
glad you're bringing that up.
If we did this with humans, that child would have 3 parents. They would have DNA from 3 parents.
Or it would be a baby developed in a lamb bag.
Right. Right.
There are synthetic uterus in development.
I'm also really curious to know what percentage the genetics they're carrying is from each parent, because it's roughly expected to be 50-50, but this is such a very specific process. I'm curious, since they were all male and they were sterile, if it's not.
Yeah, I mean, they controlled the imprinting process. So they say paternally expressed imprinted genes were overexpressed in bipaternal mice. But this is also part of the issue of some of the disorders they had. So widespread internal organ enlargement, excessive weight, open eyelids that couldn't close, protruding tongues, edema, umbilical hernias for the ones that didn't make it. So a lot of the problems that arise are a direct result of the the issues that are happening there.
Interesting. Wow. Wild.
Now, for those who are watching, we're looking at a short video of one of the offspring, not an embryo, but a pup, a very young pup that has been born. Probably looking like it's dreaming. It looks like it's trying to yarp, yarp and make vocalizations. It's like, I want friends. It looks pretty cute, actually.
You got 2 dads.
Mice 2 dads.
Yeah.
That's how that works.
Mice 2 dads.
Mice 2 dads. My last story for the night, I would like to talk about listening, thinking of those little pups that are crying out, wanting to make friends. How do they do that? Who's listening? Blair, how many animals do you know that move their ears around in response to sounds?
Pretty much all of them that have external ears, except for us.
Okay, key point, except for us. And we know that there are some people who can wiggle their ears, right? I'm one of those people, like, I have never figured out how to wiggle my ears. And usually there are some people who can really do the individual wiggling of ears, but I've seen people where they move their whole scalp to make their ears move. But it's hard. But what happened? Why don't we move our ears anymore?
Mhm, mhm.
Are
Good
there
question.
still signals? Is there something there? Is there anything left to our loss of function? This is what researchers wanted to know when they decided to look into uh, how many, whether people were actually had nervous activation of the muscles that are linked to the ear. So the auricular muscles. They decided to look at this, what they call the function of the vestigial pinna orienting system. And they hypothesized that attempts to move your vestigial pinna should be reflected in the electromagnetic generated signals of the auricular, the EMG of the auricular muscles, and can be enhanced when stimuli are presented from an out of view position as opposed to an in view position.
Mm,
So this was in Germany, and it's not a huge study, 20 adult native German speaking participants without any known neurological or cognitive deficits, 12 male, 8 female. And they were all able to hear very well everything, through the audiograms, everyone could hear. It was great. So they stuck people into a situation where they were in front of a screen and there were speakers in front of them and behind them. And then it was like a cafe situation, but they had them listening to like an audio book. And with the audio that was going on, they kind of changed the difficulty of being able to hear and where the speaker was coming from. They could move one speaker around. And so through it all, the participants' vestibular pinna orienting system was being measured and how well it responded. The results were, for the 20 people, pretty consistent through everyone, that we have muscles that go to our ears that some people have learned and trained themselves how to move their ears with, but that are consistently activated in response to people wanting to get better auditory information. So
hmm,
paying attention more as opposed to losing attention to segments of this audiobook versus the volume of the audio that was coming, like, it's hard to hear in this situation, so you're focusing on it more, trying to hear it, versus you can hear it really easily and you're not trying to hear it at all. And they did find that their participants on average were very focused on the sound, and it wasn't just hearing, it was also these muscular innervations that really don't do much at all, because they don't
mhm,
move we don't
mhm.
have pinna to move around so much
Mm-mm.
anymore, but the muscles and the innervation is still there. So I don't know, I think this
That's
is
neat.
pretty cool. Like, we have not just the vestigial pinna, these bits and pieces of our ears. We can't move them anymore, but the innervation is still there in such a way that perhaps we could get it back someday, right? Or
I mean,
maybe,
what I'm also hearing is that if I decided to get some plastic surgery to add big old ear flaps to my face, I could move them.
gosh, I have had this question for so long, right? If we wanted bat ears and like, we're like, yeah, let's innervate it. Could we?
Yeah, I don't know if you could fully do this, but you could definitely
I want German Shepherd ears. They're like boop up straight on the
yeah.
head, and then
That's Sadie's
that was.
ears. I can always tell her ear will go whoop.
But this,
Like, what do you hear?
I think your comment about plastic surgery is really spot on, and it makes me wonder a lot about, So I'm wearing earbuds right now. These earbuds
Mhm.
are small and they go directly into the ear canal, right? Whereas we have over the ear headphones that do cover the entire ear. And if we were to take like the topography of your ear into account, how could we like uniquely personalize the way that sound is sent to your ear if we took
Mm,
whole ear into account? And how much does the ear topography actually influence the frequencies uh, that are more strongly reacted to in our, what we hear more highly, the inter-individual differences in hearing, not just volume, but if my ears go back against my head, does that mean that I hear a broader range of sounds around me than somebody whose ears stick out and maybe they're much better at directional hearing, like a parabolic dish? And what does this mean for plastic surgery? What happens if you lose an ear like Van Gogh and and need it replaced? How do you reconstruct that in a way that is going to be at least the same, if not
That's
better?
interesting.
Right. And for the future, yeah. I mean, seriously, people who are like doing all sorts of body mods, right? What about different shaped ears? How will
Good
that change
question.
things? I think it's so fascinating. I love this question. Researchers, grad students, get on it. Listen up actively with all your ears. Blair, you mentioned your bat study being like mostly males.
Yes.
And other news that was looking at this week that I didn't get to reporting, but just goes right on the tail of that. Things that, of course, we should know already. And why did you publish this in IBIS this week, International Journal of Avian Science? Researchers published their study entitled "A Focus on Females Can Improve Science and Conservation." And no, this is not like female researchers. This is not female bird watchers. This is none of that. This is, hey, everybody, why aren't we looking at the female birds?
Adoy.
Hey, everybody.
Oh, I'm so sick of it.
I mean, the fact that this paper, and this is not the first time this kind of a paper has been published. But this is the first line of this paper after the abstract, I believe, is very timely. A tenet of science is to be objective and representative, but ecological research does not always consider a species' sex separately. And when it does, a selective bias in how science is conducted disproportionately obscures knowledge about females across disciplines.
Like, why is this variable? Good science is controlling variables. Why does nobody give a toot about this variable? How does this variable keep getting by? You're like, oh, I got to control for geographic location. I got to control for age. I got to control for how vocal they are. Nobody's like, oh, I should control for whether they have different sex chromosomes or not. Come on.
Money, time, it's easier. Everybody's the same, some reasons, they say in this paper here, some reasons for underreporting female birds. And oh, by the way, the last sentence of the first paragraph, this perspective focuses on birds because while sex selective or agnostic, i.e. does not distinguish sexes, knowledge is prevalent across numerous taxa, birds are among the best studied. Underreporting in females is, number one, tendency to focus on species as the default unit of study overshadows interspecific differences. Identification knowledge on female birds is sparser than on males. That's the flashy male issue, right? And the little brown birds, very often just the females, right? And three, detection and survey protocols tend to favor male birds. Flashy displays, auditory displays, calls during mating season, and etc. So, yeah, hey, it's time to focus on the ladies, on the women, on stop ignoring females. Am I talking about inclusion and diversity enough? Are we getting at equitable ecological research practices? I mean, I really want to make what we're talking about accessible to as many as possible. Oh boy, oh boy, oh boy. Thank you.
Thank you. Like, are you thanking me? Are you thanking the viewers?
For you, everyone out there. Have we done it?
We've done it.
We have done it. Everyone, thank you so much for joining us for another episode of This Week in Science. If you are feeling like you want to help science and science communication and you don't really know how, I would like to recommend that you check out the Association of Science Communicators, which disclaimer, I am on the organizing board. I'm the vice president at the Association of Science Communicators. We put on an annual conference for professional science communicators called Science Talk. It's taking place in Raleigh, North Carolina this year. And if you are a science communicator, if you know someone who wants to get into science communication, if you know science communicators, let them know about Science Talk. Science Talk is hybrid. I did that because I like, online streaming and stuff, and I want to make things accessible to as many people as possible. Our conference this year is focused on science with society. We have early bird rates until February 14th. We've extended our early bird period. And additionally, if this is not a conference for you, but you would like to help support the community of professional science communicators, we are actively trying to develop our support for travel and registration awards for professional science communicators who are experiencing financial hardships or are undergoing career changes at the moment. So, for those of you who are interested, you can donate to the Association of Science Communicators, and that will help us support more people coming together to be able to do better science communication across the country and around the world. So just throwing that out there. Disclaimer, disclaimer, disclaimer. This is one of my passions, is building this community. And anyway, ask me about it at some time, send me an email, or just go straight to the Association of Science Communicators.org and follow the links to donate or register for the conference. Love to see you there, virtually or in person, whatever works. Pepsi Cola, thanks for joining. So glad you came in. First time viewing. Woo! Okay. Yeah, thanks, Blair. All right, I'm going to stop showing this right now. And as now we're at the very, 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 very end. We've done it, right?
Very, very, yeah, very, give us some shout outs, Kiki.
It is time for the shouting outing. All right. Thank you so much to all of you who are in our chat rooms, who are watching live, who are a part of all of this that's ongoing right now, this very minute, and later watching it at a little different time shifted if you're watching it. Thanks for being in our chat room. Discord, thank you for being there. I'm sorry I'm not there tonight. I am still coming back from a full system hack that was pretty nasty this last week. So once again, I need to get access to my Discord account, and that has not gotten in there. But we know you're there, and I know that you're there because you are one of our patrons. And so the fact that you're in the Discord chatting right now, it makes me so happy. Keep doing it. Anyone who wants to join us as a patron, you can join this Discord community as well. And I will try to be there at some point when I get back in there. Blair, do you have access to Discord? Can you say hi for me in the Discord?
I just did. I said, hi, I see you. Thanks for being here.
Awesome. Vada, thank you so much for all your help with show notes and with social media. I know we haven't been easy lately with the cancellations and with other stuff, but thank you for keeping it all going for us. Identity4, thank you for recording the show. Gord, Arnlor, others who help maintain the civility in our chat rooms. Thank you for doing that. I mean, really all of you. Thank you for your civility and just being conscious people and caring people so we can all have good discussions together. Additionally, Rachel, thank you for editing the show. Can't get it out there as a podcast without you. Thank you for doing that work. And once and not finally, but yeah, we gotta say thank you to our Patreon sponsors. You ready for this, Blair?
Oh, I'm ready.
I've been slowly trying to go between. I've got a little tiny laptop here that I'm trying to make it work. I'll make it go, everyone. We'll make it happen. All right. Thank you to Lauren Gifford, Dana Lewis, Eden Mundell, Alan Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Kent Northcote, George Corris, Pierre Velazarb, John Ratnaswamy, Chris Wozniak, Vigard Shefstad, Donovan Styles, a.k.a. Don Stylo, Ali Coffin, Shubru, Sarah Forfart, Don Mundus, P.I.G., Stephen Alberon, Daryl Myshak, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Johnny Gridley, Remy Day, G. Burton, Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Riggs, John Atwood, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul Phillips, Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, Lon Makes, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Bob Caldry, Marjorie, Paul D. Disney, Patrick Pecoraro, and Tony Steele. Thank you for all your support on Patreon. All of you who support us, we really can't do this without you. If you are interested in supporting us on Patreon or through our Zazzle merchandise store, head over to TWIS.org. On next week's show,
we will be back at some time. TBD. Broadcasting, maybe live. You will find a video on YouTube and Facebook at some time after the fact. Guaranteed. We'll keep you posted. If you want to listen to us as a podcast, just search for This Week in Science where podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well. And I'm just going to keep going. For more information on anything you've heard here today, show notes and links to stories will be available on our website. That's at www.twis.org. You can also sign up for our newsletter. We love your feedback. If there's a topic you would like us to cover or address or a suggestion for an interview, please let us know on one of our social media accounts or send us an email. Just be sure to put TWIS, TWIS in the subject line, or your email will be spam filtered into the bitter receptors in my brain, but those are not the email receptors, so I will not read them. We look forward to discussing science with you again next week at some time. And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.